Cisplatin is another compound which can make cross links in the DNA. So how it was discovered that cisplatin can also damage DNA? So while examining the effects of electric currents on E. coli, a new compound, namely cis-diamine dichloroplatinum, better known as cisplatin, was found to block cell division in E. coli. Then the efforts were made to see if cisplatin would also inhibit cell division in other kinds of cells as it has inhibited in E. coli. It was revealed that cisplatin blocks the division of tumor cells. So here is the structure of cisplatin. Here you can see two chloride ligands are present and two ammonia are present, which are attached to one platinum. So after cisplatin enters the cell by passive transport or active transport, it can get entry into the cell by any way, passively or actively. It undergoes hydrolysis to produce a highly reactive and charged complex. So this cisplatin, after hydrolysis or after activation, has been converted into highly reactive complex. And this complex, being highly reactive, it coordinates to the N7 atom of either guanine or adenine base in the DNA molecule. Here you can see this is a nitrogenous base, and to the N7 this cisplatin is attached. So in this way it forms a monoadduct in the DNA. The remaining chloride ligand is displaced by hydrolysis, allowing the platinum to coordinate to a second purine base on the same or opposite strand of the double-stranded DNA. So as you know, there are two chloride ligands. After activation, one ligand was removed, and in that place this cisplatin was attached with nitrogenous base. So it formed monoadduct. But after the removal of second ligand, now it can make another linkage or another cross link with the another nitrogenous base. Here you can see this is cisplatin. It is activated, and one chloride ligand has been removed, and after this activation it binds with DNA here and here. And after the activation of second ligand, or after the removal of second ligand, this platinum can make linkage with another guanine of the same strand. So in this way this cisplatin makes the intra-strand cross-link. So in this way this cross-link is formed between the one strand of DNA, but it can also form this intercross link with another strand. So here you can see this is interstrand, example of interstrand cross-link. So this is these are two strands of DNA. This is ribose, and this is guanine attached to deoxyribose, and similarly this is deoxyribose and guanine is attached to this deoxyribose. So this cisplatin, it forms a cross link between these two guanines. So in this way it can link two strands, one and the other. So this is the formation of interstrand cross-link. As you can see here, this is one linkage and this is another. So these two linkages are on the same strand. So this is intrastrand cross-link. But In case here you can see one linkage is on this strand and the other linkage is on this strand. So here it has formed a interstrand crosslink. So in this way it can form interstrand as well as intrastrand crosslink. So in this way this cisplatin can make interstrand crosslink as well as intrastrand crosslinks. So cisplatin's cytotoxic effects appear to be due to interstrand crosslinking, which blocks the replication and transcription machinery. The intrastrand crosslink is not that detrimental. Cisplatin is a very effective chemotherapeutic agent for treating cancers of the bladder, ovaries and testicles, but unfortunately it has a number of side effects, so it cannot be used with liberty to treat cancer patients.